in case of EMB, that is eosin methylene blue, there are two kinds of dye, like I said. The first one is eosin, Y, and the second one is methylene blue. And both of these dyes, they inhibit gram-positive bacteria to grow. And when the gram-negative bacteria are growing, these EMB media, they have got lactose as carbon source. So when the gram-negative bacteria, they're using this lactose and causing the formation of acidic products, they would turn this EMB, the color of the EMB, green. So acidic products that would turn the color green. And if you grow E. coli, especially if you grow E. coli in EMB, so you're going to see a green metallic sheet like appearance on the media. So you can Google that out. It looks really nice. So this lactose fermenting bacteria on EMB, and it sort of does this with most of the fecal bacteria, and E. coli is one of the fecal bacteria. Now if we talk about McConkey, this is again selective and differential media, like I said. So they have got, the main two components are bile salts and crystal violet. So these again, the bile salts and the crystal violet, they inhibit gram-positive bacteria, and they also have lactose as a carbon source. So they can have lactose, and they also have something called neutral red. So this neutral red, this is a pH indicator. So they have lactose as well as a neutral red, which is a pH indicator. And this pH indicator, the neutral red, this sort of allows the differentiation of gram-negative bacteria based on the products that they release when they ferment lactose. So the lactose fermenting, so if lactose is getting fermented by the bacteria and there is accumulation of acidic products, this neutral red, which is the pH indicator, that would turn the color of the media, red. And E. coli is again the example which can ferment lactose and sort of give the red color in the McConkey. And apart from McConkey and EMB, we have another media which is quite frequently used in microbiology, which is the mannitol salt agar. Mannitol salt agar. So in short, we call it MSA media. Again, it is selective and differential. So the special thing about this MSA is that it has got a high salt concentration, 7.5% NaCl, and only those bacteria that can withstand or that can survive this high salt concentration. Normally bacteria do not grow in this, until and unless they're halotolerant or halophilic, they won't grow in such a high salt concentration. So if a bacteria is tolerant to this to this high salt concentration, then only it can grow. And since it has got mannitol here, the bacteria can use this mannitol as a carbon source. And again, this would form acidic products. And this has got the pH indicator, which is phenol red in the medium. And when this acidic product starts to accumulate, this phenol red pH indicator, this it turns yellow. This would turn yellow, and that's how we know it is mannitol. It is using the mannitol for its carbon source. And you can grow any kind of pathogenic staphylococcus. So Staphylococcus aureus, you can grow in this mannitol salt agar, because the staphylococci, the pathogenic staphylococci, they are resistant to this high salt concentration, and they can grow very well this high salt concentration. So that's about it. Most of the media of a microbiology lab that you're going to use. So EMB, McConkey, and mannitol. So these are some differential media which sort of differentiates between different kinds of organisms based on the things that are present in the media, such as bile salts, crystal violet from McConkey, high salt concentration for mannitol salt agar, and eosin and methylene blue for EMB. So that's about it, and I'll see you in the next one.